Hello and welcome. Today, we're going to open up what is, without a doubt, the most important instruction manual ever written. We're talking about the genetic blueprint that makes you, well, you. So let's get right into it and unpack the code of life.
You know, it all really boils down to this one big question. I mean, think about it. Every living thing is this unbelievably complex machine. So, where are the instructions? How in the world does life store and then pass down all the information it needs to build and run itself? That's the huge mystery we're diving into today.
All right, here's how we're going to tackle this. First, we'll follow the clues in a kind of scientific detective story to find the secret molecule of life. Then, we'll zoom right in on its blueprint. After that, we'll see how that blueprint actually gets used to build stuff, how it's switched on and off, and then finally, we'll pull all the way back to see the entire instruction book in all its glory.
Okay, so our story kicks off like any good mystery, right? There's a hunt for a hidden culprit. For a long, long time, scientists knew that traits were passed down. You get your eye color from your parents, that sort of thing. But they had absolutely no clue what physical thing was carrying all that information.
This case was cracked one piece at a time. The first big clue dropped back in 1928. A scientist named Griffith found this mysterious transforming factor that could literally change one type of bacteria into another. Huge. Then in 1944, Avery, MacLeod, and McCarty pretty much proved this factor was a molecule called DNA. But the final nail in the coffin came in 1952, when Hershey and Chase used viruses to show without a doubt that it was DNA and not protein that carried the instructions for life. Case closed.
And just like that, our hero is revealed, deoxyribonucleic acid. You can just call it DNA. This is it. This is the molecule of heredity, the very stuff our genes are made of. It's like the master hard drive that contains every single piece of information an organism needs to develop, to live, and to reproduce.
So now that we found our molecule, let's get a closer look. Let's zoom in and see what makes it so incredibly special. We are going inside the master blueprint to really check out its structure. You've seen this shape a million times, right? The iconic double helix, made famous by Watson and Crick. It's so elegant, it's beautiful really, and its structure, well, its structure is the key to absolutely everything it does. But what's it actually made of?
Now, before we break the helix down, we have to meet its very close cousin, RNA. Here's the easiest way to think about it. DNA is like the permanent, priceless master blueprint that's kept super safe. It's double-stranded and it uses a chemical base called thymine. RNA, on the other hand, is like a temporary, working photocopy. It's single-stranded and it uses uracil instead of thymine. This difference is going to be super important in just a minute.
So, how do you build one of these things? Honestly, just picture a twisted ladder. The two long sides of the ladder, that's what we call a sugar-phosphate backbone. The rungs of the ladder are made of 4 chemical bases, A, T, C, and G. And here's the brilliant part. They always pair up in a specific way, A with T and C with G. They're held together by these weak little things called hydrogen bonds. This simple pairing rule is the absolute secret to how DNA stores information and copies itself so perfectly.
Okay, so we have a blueprint. Awesome. But a blueprint is totally useless unless someone can actually read it and build something, right? And that brings us to our next chapter, how the cell turns all that information in DNA into real action. This whole process has a pretty grand name, the central dogma of molecular biology.
So here's the basic flow of how it all works. The DNA blueprint, it stays protected inside the cell's nucleus. Then, a process called transcription makes a temporary RNA copy of just one gene. That RNA message then travels out of the nucleus to a little cellular factory called a ribosome. And there, in a process called translation, the ribosome reads the message and builds the final product, a protein. And it's these proteins that are the real workhorses of the cell, doing everything from digesting your lunch to making your muscles move.
Let's look a little closer at that first step, transcription. Just imagine the DNA in your nucleus is like a massive, priceless reference library. You wouldn't check out the entire library just to read one page, would you? Of course not. You'd make a photocopy. And that is exactly what transcription is. The cell makes a messenger RNA, or mRNA, copy of just the specific gene it needs at that moment. Okay, now that photocopy, the mRNA, gets taken to the factory floor. That's the ribosome. And this is where translation happens. The ribosome reads the RNA's code in these 3-letter words called codons. Each codon tells it to grab one specific amino acid, which are the building blocks of proteins. The ribosome just moves down the line, reading codon by codon, grabbing the right amino acids, and linking them together in a chain until voila, you have a specific protein ready to go to work.
So this brings up a really fascinating question, right? If every single cell in your body, from a cell in your eye to a cell in your big toe, has the exact same DNA, how does an eye cell know to be an eye cell and not a toe cell? Well, the answer is all about regulation.
See, the big secret is that cells don't use all of their genes all of the time. Can you imagine? It would be total chaos, not to mention incredibly wasteful. Instead, they have these really sophisticated systems to turn genes on and off whenever they're needed. We call this control gene expression.
A classic example from bacteria, called the Lac Operon, shows this off perfectly. It's a simple, really elegant genetic switch. When there's no lactose, that's milk sugar, around for the bacteria to eat, a repressor protein just sits on the DNA and physically blocks the genes needed to digest lactose, so transcription is off. But when lactose is there, it actually binds to that repressor, changes its shape, and makes it pop right off the DNA. All of a sudden, the path is clear, the genes get transcribed, and the switch is flipped on. It's a perfect little system of supply and demand, all at the molecular level.
So far, we've been looking at single genes and these cool little switches. For our last section, let's zoom all the way out. Let's really appreciate the full, mind-boggling scale of this entire genetic library.
This entire library is called the genome. It is the complete set of DNA instructions for an organism. Not just the genes that make proteins, but all the regulatory switches and all the other sequences in between. It's the whole shebang. It is the full story of you, written in the language of A, T, C, and G.
And how big is this book? Well, the human genome has about 3 billion of these base pairs. 3 billion. To put that in perspective, if you were to type it all out in a normal font, it would fill a stack of books 200 feet high. That is a staggering amount of information packed inside every single one of your cells.
But here's the plot twist. Of those 3 billion letters, only a tiny fraction of that, like 1 to 2%, actually codes for the roughly 20,000 proteins. These are what we traditionally call genes. For the longest time, scientists called the rest of it junk DNA. But we now know that a lot of it is involved in that supercritical regulation we talked about. All those on and off switches that orchestrate the entire symphony of life.
So let's just do a quick recap. DNA is the master molecule of heredity. The information inside it flows from the DNA blueprint to an RNA message to a final protein product. The elegant double helix structure with its specific base pairing is the absolute key to its function. And this whole process is precisely controlled by gene regulation, all contained within the complete instruction book we call the genome. For the first time in human history, our own genetic blueprint is basically an open book. We can read it, we can understand it, we can even start to edit it. The science has handed us the pen. So the real question that leaves us with is, what will we as a species choose to write in the next chapter of our own story?